Hello, my name is Anastasia Kokoska, and today I will be discussing exam question 9 related to positron emission tomography, structural magnetic resonance imaging, functional magnetic resonance imaging, and the findings of Nikos Logothetis. So to start, I will be discussing PET scans. The PET is a scanner that uses tracers to measure blood flow in the brain. The patient is injected with a radioactive tracer that attaches to glucose, which is the main fuel source in the brain. And by using the PET scanner, the researchers can then actually pick up gamma ray signals from the brain tissue that is using more glucose, and that is thus more saturated with the tracer. So researchers use this to see how the brain is functioning and whether there is any disease or injury present in the brain tissue. So basically, the PET scanner picks up the signal given off by the decaying isotopes in the tracer that was injected into the body. Sometimes they'll have the patient take a pill as well. The signal is then processed by a computer and is used to create 3D images of the brain slices. Next, I'm going to talk about fMRIs. The fMRI is a physiological, non-invasive technique that is used to measure blood oxygen levels and analyze the brain's functions. As well, researchers will use this to detect possible injuries and diseases in the patient. Additionally, the foundation of this technique is that neural activity is correlated to more blood flow in that specific region of the brain that is being activated. So researchers take advantage of this physiology to detect any brain issues and even conduct brain mapping. So fMRIs also use the bold response, which is the blood oxygen level dependent response. This response is used to measure the change in blood flow in specific areas to map out neural activity. It's similar to the PET scan in that it is based on neural activity being related to changes in blood flow in different areas. However, the fMRI is way safer than the PET scan because the PET scan, as I said, uses radioactive isotopes. So as well, the picture in the bottom right corner was one that I grabbed from page 88 of the textbook. It's an fMRI scan from a subject that was looking at pictures. The imaging suggests occipital lobe activation when presented with visual stimuli, which makes sense because the occipital lobe is responsible for processing visual stimuli in our brains. But this is just one example of fMRI scans being used in cognitive science research. But a lot of the time, researchers will have their patients perform a specific task while they're in the machine and then analyze their fMRI results. The regular structural MRI is an anatomical non-invasive technique that applies electromagnets to measure average tissue density. It differs from the fMRI because while the fMRI measures brain activity, the MRI looks for anatomy and structure. It's used by doctors and researchers to look at the shapes and sizes of different brain structures and can be used on both living and dead brains for study. Overall, PET, MRI, and fMRI all use the same machine structure of a narrow tube that the patient is placed into, but each analyzes the brain in a different way. Finally, I'm going to talk about Logothetis and the bold signal. Nikos Logothetis is a researcher in neurophysiological recordings who simultaneously utilized an fMRI scanner and electrodes in his research with macaque monkeys. He actually found that the bold response is correlated with local field potentials. So this graph here was taken from this week's PowerPoint. You can see that the blue line of the bold signal correlates to the brown local field potential line, and this is what Logothetis found in his studies. So, thank you.